everyone, I'm Annie Zhao, and today I'm going to be presenting my comparative study for indoor plants for PM2.5 absorption. So I am a senior at San Marino High School, California, and attached is my email if you have any questions. So first, why do we want to investigate the plant's potential to capture indoor PM2.5? So, indoor PM2.5 is known to cause asthma, wheeze, and long-term effects such as lung cancer, Alzheimer's, and even Parkinson's disease. And as well, people spend 90% of their time indoors, and a smoke-free household could inhale 70% less PM2.5 than smoking households do, which is why we're going to be using cigarette smoke simulated in this experiment. And why using plants for mitigation? Well, an old study by Wolverton et al., published by NASA in 1984, was the first to analyze efficiency of indoor pot plants for air quality improvement. And in this study, we'll be selecting plants with distinctive features such as leaf area, morphology, and stomatal density, and we hope to establish some type of association between any of these traits and PM2.5 capture. For methodology, I selected the 6 plants as following: Boston fern, spider plant, peace lily, English ivy, aloe vera, and rubber plants. These are the plants that are also prevalent in other research articles, so they're worth investigating. They were purchased from a local nursery, and they were all baby-sized plant pots, and 3 pots per species were used to reduce error. For my semi-airtight chambers, I modified them using 4 18-liter water jugs made out of PET, which is a type of plastic. The specific dimensions are 26 cm in diameter and 48 cm in height. All of these 4 jugs were cut open at the halfway point so that the plants were placed inside. And another circular opening of 0.5 cm in diameter was cut so I can fit the PM2.5 monitor in. And for the monitor, I used Temtop M2000C. It is a household device readily available to purchase by anybody. And how that works is it basically graphically scans the airborne particles, then transmits the numerical data in micrograms per meters cubed. And for data collection, I injected cigarette smoke into the chamber from the lid while the PM monitor would be plugged into the hole, and that would measure the beginning concentration. And after 6 hours, we would measure the final concentration of the PM2.5, which is cigarette smoke. 4 to 6 valid trials were run on each species, and 3 leakage trials per jug were also conducted. So a specific image for the experimental setup is as is. For data methodology, after all these data were collected, I calibrated them into hourly absorption rate of PM2.5 for each plant and trial. The average rates were then plotted along with the standard deviation on Excel and Google Sheets. As for my results, this is figure 1. So this is a 5-number summary of all the plant types that I've experimented with. And as you can see, English ivy ranks the highest for median hourly absorption rate at 15.78%, followed by peace lily at 15.67%, and lastly, Boston fern at 14.28%. However, these absorption rates are only nominal. And to account for leakage, this is my leakage table. So I did 3 trials per box, so that averages to 12 trials. And since their standard deviations overlap, I then took an average of the 4 averages shown here, and I subtracted them from figure 1 to get figure 3, which is our real average hourly absorption rate of PM2.5. And as you can see, English ivy, of course, still ranks first at 6.21%, followed by peace lily at 6%, and lastly, aloe vera at 4.47%. And as well, since we cannot assume a linear relationship, I have went ahead and I have presented them over a 6-hour average rates, which is a closest representation of the raw data. And the percentages are as followed. English ivy is at 37.38%, followed by peace lily at 36.10%, and lastly, aloe vera at 26.93%. So overall, the effectiveness of ornamental plants in PM2.5 regulation is that it is a low-cost option available for households located in highly polluted cities where outdoor ventilation and air purifiers are not a viable option. So the actual hourly absorption rate ranges from 4% to 6%, which is pretty insignificant, but over 6 hours, according to the English ivy data, it reduces 37.77%, starting from a very hazardous level, which is according to the US EPA, 350 to 500 micrograms per meter squared, to good and moderate, which is a significant reduction. Trichome density was also explored. It's an evolutionary trait prominent in plants such as succulents, cacti, and aloe vera, so that they can repel against herbivory and help reduce water loss in arid and hot conditions. And out of the 6 plants, since aloe vera is the only one to adapt to live under such climates, it can be assumed that it has a relatively high trichome density. But that contradicts its low PM2.5 absorption. So we have eliminated trichome density as a factor for PM2.5 absorption. However, that is not the same for leaf surface area. So from observation alone, the plants with the highest leaf surface area used in the studies are spider plant, English ivy, and peace lily. And all 3 of them actually had the highest PM2.5 absorption in this study. So it is very evident that this is a trait that it has a positive association with PM2.5 absorption. Similarly, with stomatal density, English ivy has a very high stomatal density at 125 to 240 per millimeter squared. Peace lily at 30 on the abaxal and 4 on the adaxial sides. So those are the different sides of the leaves. And at a much lower value, aloe vera only has 9 per millimeter squared, and 8 on the adaxial side. So it seems that stomatal density might also have a positive effect on the PM2.5 absorption. And last one is plant morphology. So by visual observations again, English ivy has a medium to medium fine furry leaf texture. Peace lilies, on the other hand, they have large glossy leaves that have sunken green veins that adds to its leaf topography. And then coming in third for the absorption, spider plants have a narrow strap-shaped leaves, and each one has a distinct channel down the middle, and those traits all possibly aid in the capturing of PM2.5. Limitation of the study obviously includes the leakage as well as ventilation and atmospheric PM2.5 levels during the time of the experiment. And lack of professional equipment also magnifies these variables even more. There's also not enough data to generate a complete absorption curve, and also a lack of numerical measurements for trichome and stomatal density that are required to link the validity of the absorption rates to the traits that each plant is specialized in. And for my conclusions, despite the confounding variables that largened the error of my study, English ivy's effective PM2.5 capture rate still managed to exceed other plants considerably. And since it ranks really high in terms of stomatal density, rough leaf morphology, these rates are very likely to be associated with the regulation of PM2.5. Further research could focus on individually testing any of these characteristics, especially stomatal gas exchange, as a viable mechanism for PM2.5 uptake. English ivy and peace lily are relatively effective at removing PM2.5 from a potent concentration and over a long period of time, such as the 6 hours used in the study. Additional absorption curve can help solidify the logistical relationship between the PM2.5 reduction and the plant's ability to metabolize over time, which is also a crucial significator of plant health under the effects of PM2.5 and air pollution in general. These are my references that are used in my paper, and thank you so much for listening.